Hi everyone, I'm Deepak Tanwar and I'm a master student in Dr. Sarah Kimmins lab and Dr. Jeff's CR lab. He's my co-supervisor. My role in is to develop a bioinformatics analysis pipeline for sperm chipseq data. So I will start with introducing epigenetics. So it is an additional layer over genetic information which get inherited to offsprings without actually changing DNA sequence. So epigenetic modifications or epigenetic marks could be thought similar to punctuation marks in English. So if you say this, you actually won't be able to understand what it is actually, if it's a word or a sentence. Because there are no punctuation marks, there is no spacing between the words. It actually just says like bioinformaticians are not antisocial, we are just genome friendly. So if you say that if this is the DNA sequence, then epigenetic marks are basically the punctuation marks. Or to be more generic, if this book is a DNA sequence, then the sticky notes could be thought of epigenetic modification or epigenetic marks. These epigenetic marks are being inherited by our offsprings by a mechanism called epigenetic inheritance. So when a human is exposed to environment, the epigenetic modifications are induced into somatic cells. This information is then transmitted to germline via circulating microRNAs. After that, this information is carried out to oocytes and a phenotypic trait is inherited by an unexposed offspring. So we are studying basically in histone methylation in our lab. So you are seeing a structure of nucleosome here, which is a unit of chromatin by how DNA is packed into chromosomes. So a nucleosome is made up of 4 histone proteins, and they have these 4 tails and terminal tails coming out of the nucleosome structure. These epigenetic modifications occurs on these tails, and there could be many epigenetic modifications which could occur like acetylation, methylation, phosphorylation. So epigenetic marks can play 2 major roles. One is activating a gene expression or deactivating it. And we are studying STK4 trimethyl because this is present in the promoter region and responsible for activating gene. The function of sperm histone modification are not known in epigenetic inheritance yet. And Hamoud and all, they shows that nucleosomes are retained at promoters, and some of them are actually marked by STK4 trimethyl. Also, the genes which are marked by STK4 trimethyl have functioning in cell process, metabolism, or even in development. So sperm epigenome is unique because it has 85% of DNA binding histones which are being converted to replaced by proteamines. So actually we have only 15% of histones left in sperm in human or 1% in mice. So and also as the chromatin structure of sperm is unique, we do require a specialized bioinformatics pipeline. The method by which we study, I think everyone knows about it, is CHIP. So we extract the nucleosome, we extract the chromatin and then fragment it into nucleosomes. Then the nucleosomes are then targeted by antibody, and the process is called immunoprecipitation. The DNA is then extracted after purification and then sent to sequencing center. And from there, my research focus begins. So we have sperm chipseq data from sperm semen samples from Great Fertility Clinic, where we have the reference population from Canada, and also samples from fertile and infertile individuals. According to WHO, World Health Organization have certain specifications for a sperm to be called as fertile, and even they met all the parameters. The infertile samples, still the reason for the infertility is unknown, and we are trying to see if we can address this by targeting sperm by STK4 trimethyl. So the question we are asking is if STK4 trimethyl is associated with idiopathic infertility. For this, I have developed a ChIP-seq pipeline. So basically, when you send your data to sequencing center, you obtain a file called BAM file, in which the reads of your data are aligned to reference genome of their choice. So whenever you get data, the first step you perform is quality check. And for that, you have to go through a number of steps here. And after that, the next thing you do is peak calling, which is major step in this field and challenging. And This is basically the basic pipeline which you find everywhere, but we have also trying to integrate the statistical analysis part here in the pipeline, which would be very helpful for researchers. Thank you.